Yeah, so I'm an associate professor at the Department of Psychiatry here at McGill University in Montreal. I'm I'm a pediatrician by training and with PhD in neuroscience, and I have my lab works with the long-term effects of childhood adversity or early life stress, how that affects neurodevelopment, behavior, risk for disease. And we use different types of approaches to try to understand these effects. Like we use rodent models, also large data sets of humans, genomic studies, neuroimaging studies, trying to address this question, how early life stress modifies development and risk for disease in the long term. Yeah, that's the broad question of the lab, and I think that we got together because of your interest in the expression-based polygenic score, which was a tool that we created to address this question, to study this. The tool ended up being applicable to many different other questions, but it was originally created to test this and to do this bridge between what we found in the animal models, that we can really explore the molecular mechanisms associated with early life stress, what happens acutely, what happens in the long term, and children and humans that we can't really measure things in the brain. We can't really study, molecular mechanisms in detail with the same level of detail. So we started using genomics to try to do this bridge and study individual variations in genes initially. But you know this well, that genes do not work alone, do not work in isolation. They work together in coordinated networks, and they help each other in biological processes. So we started to understand that we needed to go beyond understanding the effect of a single gene or variation in a single molecule, to understand variation in several molecules working together in networks. So yeah, that was the basic idea of first try to identify a co-expression network that was involved in a biological process, and then use this information to inform the development of a polygenic marker to understand individual differences.
And to, how in the end, how happy are you with this? Do you think that it's basically a good tool now, or is there a lot of room for development of new markers?
No, I think it's a nice tool. There's a lot of room for creation from here, I think. We started, for example, we started by simply selecting genes that were co-expressed with a specific gene, like an anchor. So, for example, one of our first papers was, because I work a lot with how metabolic signals act in the brain and modify neurotransmission. So I'm always very interested in how insulin, for example, produced by the pancreas, is important for peripheral glucose metabolism, enters the brain and modifies neurotransmission. So we wanted to understand individual variation in the expression of the insulin receptor network. So we identified all the genes that are co-expressed with the insulin receptor in a specific brain region, the mesocortical limbic system, and identified this network and created a polygenic score that reflected variation in the expression of this network in this brain region. We did this in different brain regions, showing that the networks are brain region specific, like gene expression is brain region specific. But more recently, we have been, we continue doing this anchoring and trying to find the co-expression network of a specific gene, But you can also find networks that are associated with an exposure. For example, we have models of early life adversity, early life stress, and you can identify gene networks that are linked to the exposure, right? By looking at gene expression, genome-wide gene expression, and doing analysis like the WGCNA, where you identify co-expression modules associated with this early life adversity exposure. So when you do that, you have a network that is nameless. It's just associated with your exposure. There's no anchor gene that you use to start your process with. So this was a different way to identify co-expression networks that were relevant to any sort of condition. We are interested in early life adversity, but it could be, in response to a drug or a certain phenotype that you identify in your model, or in human tissue too. For example, we are still working in this project where another scientist identified genes that in the blood were associated with early life adversity or early life trauma. So they did RNA sequencing in the blood, identifying networks that are linked to trauma. And then you can use this to inform the development of a polygenic score. What you're measuring in the end is really individual variations in this group of molecules or this network or these biological processes, and how people vary on this, and link this to disease or risk for disease.
Was hoping to maybe say a couple of words about why I thought that you two talking would be useful, and then I'll be quiet and listen to both of you. So I attended a presentation by Michael Meany, your colleague, esteemed colleague in New York City at the Congress of Neuropsychoanalysis, and he gave a talk. The whole Congress was about development and, of course, human development, and he talked about the profound influence of early childhood adversity or enrichment for the development of a child. And he presented this work that you're mentioning. So, we essentially have a biomarker, perhaps one of them, and he talked about the EPGS score with glucocorticoid receptors in hippocampus. And the very interesting result was, to be metaphoric, is the sort of dandelion children and orchid children, where there were these very sensitive children with this particular genetic profile, who, if they're exposed to childhood adversity, they would become susceptible, but if they're exposed to enrichment, they would become resilient. And more interesting, Michael was able to articulate the entire causal chain of events of what happens, in animal research when a rat is licking the pups, what's going on, all the way through with demethylation and other mechanisms of why, the CRF expression and the whole HPX is functioning, why this actually worked this way. So there's the entire story, which is coherent and makes sense. And when I mentioned this to Michael, with your work with xenobots and anthrobots, and Michael said, well, we can test this. We can try to create enrichment environment for xenobots and anthrobots, or adverse environment. And they're made from xenobots from frog skin cells and heart cells, and anthrobots from cadavers, which is epithelial tissue from trachea, I think. But I thought that, is there any possibility to do the EPGS as just data point? And we don't know what's going to come up from it. But if we were to look at the donor and look at the EPGS and then see what's going on with the anthrobot composed with a certain profile of EPGS, that was one of the ideas. And the second idea, I think that you both share this precious freezer, which is mighty 80 Celsius, where you have a lot of neuronal tissues in McGill at Ludmer Center. And I don't know if the neuron survives freezing and thawing, but the glial cells might. And so You know, I was thinking, what if an anthrobot is composed, if it can be composed in a stable way from these glial cells that where you do so. But I was thinking, being informed by your work with EPGS may allow us to get additional data. Michael mentioned the interesting experiments with tadpoles, where when exposed to GABA, you saw a bimodal distribution where these ones did this and those ones did that. And I immediately thought of your work. Then what if these are susceptible and these are resilient? But these were just some preliminary thoughts, and but I am, yeah.
Yeah, it's very interesting stuff, and with this, I think there's a lot of areas of intersection. Could I ask a couple of questions, and then we can talk about experiments and bots and stuff like that? When you said they are specific to regions of the brain, does that mean that these are typically not expressed outside the brain at all? Are all of these things brain specific, or can you find them in other tissues?
You can find in other tissues. It's just that because we start the whole thing with gene expression data, and gene expression is tissue specific. So when you do that, you identify, for example, for the insulin receptor, we did this in the striatum PFC, in the hippocampus. And when you try to identify the co-expression network for the insulin receptor in these different regions, you find completely different networks, because the function and the expression will be dependent on the region. But we have created EPRS starting from peripheral tissues too, from the liver, from the adipose tissue, from the pancreas more recently. If you enter this into a gene and you do in different regions, every time the network is very different. It's tissue specific.
And okay, and when you've defined the network, by any chance have you or anybody else parameterized the network into a network model that you can actually run and execute, and you can, there are numbers on the degree to which the different genes turn each other on and off and so on. Is there an actual GRN model for something like this?
Yeah, not like that. It is more simple. So we just see the relationship between each one of the genes, like if they are co-expressed or co-repressed.
Yeah.
And we consider this into the polygenic score calculation. So for example, if one gene inhibits the other gene, we switch the sign on the calculation, so that we know at the end, when you have the sum of all the values for all the genes, you know that a higher score will reflect a higher capacity to express that network, and a lower score, a lower capacity.
The reason I ask is that if there was such a model, and I mean it's a lot of work, but you can infer GRNs from data, right? So in theory, we could do this. We have a bunch of work over the last few years looking at how gene regulatory networks themselves store memories. And the latest, which isn't published yet, but there's a preprint looking at how certain stimuli can delete memories. They can make them forget specific memories. And I think it would be very interesting to analyze that network in particular to see, I mean, maybe the memory is somewhere else. That's always possible, but just to ask, that network alone, how much of the stressors are actually stored in the dynamic systems memory of just that network alone. And when we infer stimuli, what we do is not look for gene therapy or things like that, but stimuli that can be delivered by drug, pulsed pharmaceutical interventions and so on, to wipe certain kinds of memories from that network, and what the somatic and then the psychological effects on the body would be of doing that.
Yeah, for sure. And I think that applying more elaborate or newer mathematical models to infer the design of the network or the components of the network would be fantastic. We have so far been very concrete and just, using RNA sequencing, identifying genes that are correlated, and that's it. But yeah, I agree, using this other type, there are so many ways to identify networks, right? So for example, Some students are now working not with gene co-expression, but protein-protein interaction and creating a network from there. Other types of like co-localization, so selecting only proteins that are in the same microspace there. Most of the work we've done so far is really based on co-expression from RNA sequencing and the basic relationship that you find from these values from crude measures of gene expression. Yeah.
Interesting. Okay. I do you know whether this has anybody tried this in vitro? Has this been have any of these things been measured in cells that are and I'm not sure that any kind of in vitro culture is low stress as far as the cells are concerned, actually, but certainly we can crank up the stress if we wanted to. So the question is, have you guys done any of this in vitro?
Yeah, we're starting to do these exactly as you were saying too, Alexey, like to do these validation work when you have the genotype that you can calculate the score, but you can also measure the expression and see how much the score is linked to actual measure of gene expression. This work is at the very beginning, so yeah, it would be fascinating to do this either using postmortem tissue, that's what we're doing in collaboration here with the brain bank, but also using your models as well. It would be a nice validation for the method too, for sure.
I think it'd be very, kind of like what Alexey was saying. I think it's very interesting that when we make our anthrobots, occasionally they come from cadavers, but often they come from living patients who come and donate, they do tracheal biopsies and they just donate the cells, right? So many times we do have a living patient. And I don't know, I've not done human studies before, so I have no idea how difficult or easy this actually is, but presumably we could ask some of them for some sort of a psychological survey, or I don't know how you guys determine it, but try to, I've wanted to do that anyway, to basically do behavioral testing on the bots to see if they correlate at all with the psychological properties of the donor, right? And so how much do the bots reflect? At the very least, certain, it would be interesting to look at certain kinds of addictions and things like that. It should be not too hard, but really something subtle. So what do you think, how doable and what might be the cost of such a thing to do a test, to do a project where we take some reasonable number of patients and get some kind of a profiling on them, and then get the samples and grow the bots and do analysis? What do you think?
Yeah. No, I think for us to do this investigation, what we would need from the EPRS side would be the genotype of the participants and gene expression level, like RNA sequencing. I know that you already do, like transcriptomics.
Yeah, that you could do because if you're going to do that anyway, that you could do on the cells, right? So actually, if that's the case, you don't need to involve really the patient at all, because you're getting, they're already donating the cells. You get the cells, you do your genotype, you do your transcriptomics, and then we do we make our plots and profile them. So that may be not so hard, yeah?
Yeah, I read your papers, and I think that even data that is existing could be used for an initial analysis, right? I don't know. I don't know how much you can access in terms of genotype from the materials, but I'm sure you have.
It's not hard. We can nowadays genotyping them is not hard, and we haven't. You're right. We've published at least a transcriptome. Yeah, interesting. Very interesting. Okay. Well, that's probably worth trying.
And yes, of course, starting from human cells is fascinating, but a lot of the work that we do starts from models as well. So even the bots that you have that are created from cells, like animal cells, also could work.
Yeah, we have. So unfortunately, we don't have any mouse bots. We probably could make them. It's just we don't have the, we don't have a source, an easy source for the tracheal epithelial cells. Probably we could get them. I'm not sure what that would entail. But the other thing we do have is frog. So this is frog embryo epithelial cells, which make xenobots. There we have the adult animals. We have the cells. Yeah. Could do that, and we can also, as Alexey was saying, we do want to start looking at environmental features. So we can definitely do enrichment. We can it's easy to do some sort of adversity. We have now we've recently published that they're sensitive to vibration, so acoustic kinds of things. We're playing with light. Not clear to me yet how much they care about light, but acoustics, they definitely care about. Electrical signals in the water, they care about. Chemical signals, we now know, this is not published yet, but we know they can sense various things in the water. So there's kind of a wide range. Also, we can make mazes. We can 3D print mazes for them to play with. And they also, I mean, this is something I wanted to do. We're gonna this is not hard. We wanted to do this is bots that so if you provide loose epithelial cells in the environment for the xenobots, what they do is they run around and they kind of collect them into piles and they polish those piles, and those piles become the next generation of bots. So it's a really weird kind of, like, basically we've made it impossible for the xenobots to reproduce in the normal way because they don't have any of the frog organs, but they found a different way of making copies of themselves, which is just wild. But what I'm interested is in comparing bots that had a chance to do that and bots that never had a chance to do that. Like, what are the differences, right? Do they, is there some long-term consequence of, yes, I've done kinematic self-replication, I've made copies of myself, I am, happier, fulfilled, more stressed. I don't know what the answer would be, but like, do they, is there some marker of that do they know that this is what they've done, as it versus, you could do controls with passive beads or nothing and things like that.
Can you vary nutrition too?
We can. Here's the deal. Just hang on one second. I'm just taking some notes here. Yeah, you can. So normally what powers them is maternal yolk. So the cells are made of they're from a fairly young frog embryo, which means they still have a lot of yolk, and this is what they're powered by. They live not much longer than a week, if based on that. But you can put things in the water, that the nutrients in the water that they will take up. Our record is 84 days to keep a bot alive. It's actually extremely interesting because it started turning into something. I have no idea what it's turning into. It's very odd, like, it looks very weird. It becomes this like long transparent thing with a black spot on one end, which I think might have been an eye spot. I don't know exactly. The problem is we don't have a lot of these because it's a funny practical reason. The nutrition that we use for them, the bacteria so much that they get immediately overrun with bacteria. So keep them clean while continuing to feed them is very hard. We've tried all the obvious stuff. We've tried antibiotics and titering them out. It's really hard. We're still fighting it because I would very much like to keep them going and find out what happens 6 months, 6 months later, what do they turn into. So yes, we can feed them in the short term, but we're still fighting this bacterial issue.
And can you stress them by providing less nutrition, or they die?
No, I think they're pretty stressed when they start to run out of nutrients. And so you can modulate that. There's a million ways to stress them. I mean, you can give them heat shock, you can put electric shock in the water. Pretty much everything hates, interesting, every life form hates electric shock. So you can shock them. The vibrations, we're still trying to figure out how they feel about the vibrations. My guess is that we're going to find some kinds of frequency/amplitude combinations that are neutral in terms of, okay, this is signal, I can interpret this, versus this is really annoying because it's shaking everything, right? So probably we can find some kind of version that is that's annoying to them.
That's interesting, super interesting. I ask about restricting nutrition because that's one of the models that I work a lot with in rodents. So we have worked in identifying networks that respond to this kind of stress during development, not development, prenatal development. So prenatal food restriction to the dam, and then this has long-term consequences for the health of that animal. We see resemblances with low birth weight effects in humans, so that's why the model is interesting for us. But working with a type of stressors that we know well in a model may help because maybe we find some similarities in terms of networks and things like that.
I may make a quick comment on that part, Patricia. Michael and I met Frank Putnam, who's a traumatologist and specialist in DID, a psychiatrist, and he was one of the authors of a study, multi-generational study of trauma, very sad cases of women who were abused in early childhood, typically complex trauma. And what he's saw is obesity rates were very high, to talk about insulin, and of course we're talking about mesocortical limbic dopaminergic system, exactly what you study with the DAT1 transporter, and also a faster aging. So we talked about that. And so where this work gives hope to human clinical work is if there are genetic studies and you see the EPGS score suggesting susceptibility, then for that child specifically, early intervention, environmental intervention would make so much more difference. So this is actionable while the genetic profile cannot be changed, but this can be. So we can eventually have either therapeutics or suggested interventions for. So this is where this work can go, but it all tracks, so I'm saying that has application to clinical work, and I don't know, but in Frank's paper he mentioned specifically this demethylation region that you mentioned, you and Michael Meany. He mentions exactly that, so it's known. Your work is known in the trauma community. So yeah.
That's great. Yeah. What's nice about this, Alexey, that you're saying is that coming back to the cost of the experiment and all that, is that if you have the genotype and transcriptomics, you can actually explore whatever network, and it's not one shot. You can calculate all kinds of polygenic scores and different types of networks that you identify that are relevant. So it's work that can be investigated in many ways and answer different hypotheses if you have the data.
That gives me an idea what you were just saying about the transgenerational business. I wonder if we can see evidence of that in the kinematic self-replication of the bots. Like, is that considered would that work as a transgenerational thing? And I mean, it seems fairly simple. All they do is push the cells together, but maybe there are different subtle ways of doing that bots that had been traumatized would do it differently, and maybe the second and third generations would. I think it would be very interesting to look at potential transgenerational effects in kinematic self-replications.
And I had a simpler thought, the two kinds of fitness you mentioned, they live on average about 3 weeks, and the regeneration doesn't go forward other than 3 generations. So it's, grandfather or grandmother, daughter, and then granddaughter, and that's it. So you could look into, again, the environmental applications early on. Let's say you just composed a xenobot, and there's enriched environment, and see if they live a week longer or if maybe their regenerative fitness, goes one more generation. I don't know. It's a wild kind of guess, but in addition to a very concrete measurement of susceptibility and resilience, where you apply a certain number of stress and you see the reaction, where high reaction could mean susceptibility, low reaction could be in resilience. This can be measured but also you can look into sort of life fitness and regeneration fitness.
Yeah. No, that's very interesting. I have kind of a weird philosophical question for both of you, I guess, from your field. Obviously there is such a thing as, for sure, like terrible trauma and all of that. But on the other end, Okay, what is the best case scenario? I mean, if you ask people, older people who have had kids, you say, how do I prevent from screwing up my kids? They'll say, well, you can't. There's just different ways to do it. But we're talking about the very minimal, sort of the best possible scenario, right? So my question is, what do you see as, if we took care of all the, the social stressors, the resource stress, you take care of all that, you're in an ideal environment. Still, there are going to be some things, right? Kids are going to find out about certain things that are stressful to find out about and whatever. Like, what is the actual least amount of stress? And do you actually think that there is a least amount that's too little? Like, what does that look like? What does the left end of that spectrum look like?
Do you want to say something, Alexey?
Sure. Yeah, I guess I'll be maybe a little speculative, not very precise to your very creative question, but we talk about the window of tolerance again, so that when stress is, this is the Yerkes-Dodson law, a pretty old thing, which has been refined, but sort of when stress is too much, we're moving toward the trauma category, and that doesn't improve any resilience. When stress is too little, then there's no motivation to grow and not, to apply kind of effort, there's sort of, and so in the middle, when there's enough stress within the window of tolerance, then there's certain growth and development. And so that there's a dose dependency there. But also, if we talk about sort of ideal environment, maybe I'm wearing off a little bit, but in humans, first of all, the programming starts in during pregnancy, where the mom's heart rate goes up, the fetus heart rate goes up. And so when mom is very stressed out during pregnancy, then the stress regulation network in the child starts getting programmed right there. And then there's birth. And since birth, we have, of course, the nutritional things and all the biological things, but we have the attachment style, which gets formed from about 6 to 18 months of age. And these are interactions between the child and the primary caregiver. And Beatrice Beebe's work is relevant here, that just, what exactly happens between the baby and the mom, pre-verbal baby, and the whole Goo Gaga dance, and how exactly they interact with each other has profound influence on the whole life forward. The highest correlation we have of psychopathology is disorganized attachment style. So these children, and when mom is very traumatized, it is objectively very hard for her to do that with a baby. Not to blame the mom in any way, but this is one of the mechanisms of, if you wish, transmission. Not genetic, really. It's certain what happens in the interaction, what happens in pregnancy. And so then that lays the foundation. And from there forward, you can see what happens to the child in the environment. But then there's parenting, and again together, and we call it zone of proximal development, where a parent and the child, when they do things together, and the stress is gradually increasing as the child matures, then we have optimal development. When the child is thrown into the ocean, says learn how to swim, that's not optimal. Or when there isn't stress whatsoever, and the parent is helicoptering, and the child doesn't get any frustration, there's no development. So it's a very delicate dance.
Yeah, I agree. Everything is a balance, right? If you think, as you just said, if you think of, the most precious thing, maternal care, that Michael studied so much, it's so special, very important. But then you can always go to overprotection, and helicopter parents, as you're saying, then it's not good. Same thing about nutrition. If you lack nutrition, awful for development. The individual can die from this, but overnutrition is also not good. So yeah, the balance is the key, and not easy to say what's optimal there. Yeah.
Do you think that are there systems in the body that react to insufficient stress? So let's say in the kind of extreme helicoptering thing, is there some kind of a system that reacts as, again, paradoxically, another kind of stressor is, and there's not enough stress, we're not growing, whatever. Is there anything, or there's only sensors for the kind of the other side of the spectrum?
It's interesting you're saying this, because we're writing about this just now, but I think that stress is inevitable during development, right? As much as even the overprotective parents, they can't really avoid this. But there are very well established and controlled systems for stress responses. They are adaptive. They are there to help the individual survive. and they are necessary. So for example, inflammation is one of the systems that are part of the stress response, and we need that. We need the system to be trained, exposed, trained, so that we can face infections and fight disease. So there's a level of stress that is inevitable, is part of development, but is adaptive too, and the system is ready to face it, to learn with this, and to grow as part of development. I don't know biologically, like, is there a biological basis for this lack of stress is bad. I don't know that. I can understand this from a psychiatric perspective. Biologically speaking, I think it's more about this, like stress is there, is part of our nature to explore, and we need to go out there and survive. And the system is like, it's ready to, the stress system is ready to face these things. We'll learn from these experiences, and this is part of neural development. Yeah.
And maybe add a psychoanalytic perspective. I mean, Freud said ego grows in frustration. When there is no frustration, then there's sort of underdevelopment. And, Winnicott added to that, he had this beautiful concept called good enough mothering, which is, just gradually letting go, because babies are completely dependent and adults are completely independent. And our job as the baby matures and grows and develops capacity, to give them space to grow into and to let go. So when we don't let go, when we helicopter, then they're underdeveloped, which perhaps in the contemporary terms means lower allostatic complexity. So then when they leave the world, the reality of the world hits them hard. It's a rude awakening because they don't have enough allostatic complexity to handle the unpredictability and the other things. While if they're gradually exposed to increasing amounts of stress within the window of tolerance, then they are become optimally prepared for the environment to go on their own and adapt.
I was thinking a little bit back to those, the rat paradise experiments. Remember this stuff? Where they made somebody, I forget, unfortunately, I forget who did it, but somebody basically created a paradise for rats where there was as much space as you wanted, as much food as you wanted, as much nesting material as you wanted. Like, there's just absolutely nothing to worry about, right? And he documented some very interesting phenotypes of what happens in the long term, and that they ended up. You probably know this better than I do. Do you know what I'm talking about? If probably butcher this, but as I recall, what happened was, you ended up with this interesting phenotype of these male rats that would spend basically all day grooming themselves and making themselves beautiful, and they lost all interest in mating and doing anything else. They just kind of, and so it was very interesting. It was a new phenotype that behavioral phenotype that came out, where they just had absolutely nothing else to worry about, and this is sort of what they focused on. And so I wonder, like, under those circumstances, is there any stress being activated, or is there some kind of a group phenomenon where the individual animals are totally happy because they have nothing to worry about, but as a group there's something detectable going on, or something? I don't know. This is what I think about into the future, as robotics and regenerative medicine and anti-aging and this stuff comes along. What does that look like, and what does the transition look like? It seems to me that a mature society, out there in the universe, if you visit a mature civilization somewhere, presumably they're not still worried about all the dumb stuff that we have to worry about. But then what does that look like, right? What does that look like psychologically? What is the level of stress when there really isn't any stress about food or resources or anything else? What if anything should there be stress about, right? Well, like, we've never had to deal with that because we've had plenty of stress that's sort of forced on us by scarcity of environment, but eventually presumably we'll get past that. Then what? So I think it's interesting to think about.
Yeah, for sure. Super interesting to think about these things. Yeah. And the immediate thing that I think about when you say this is relationships, right? Even if you fix all the scarcity in terms of nutrition or environmental stimuli and exercise, metabolic health, there's still we're still social individuals, and relationships can be complicated. How can we fix them, like, ideally?
Have you seen some of those robots they're making? I mean, this is a people are addressing this. People are on top of this problem, right? Like this is one of those things that I think we're also going to have to deal with. If you want a simpler relationship, I think you'll be able to get one. Um, yeah. And if it's too simple, I think you'll be able to go in the apps and dial up. I need a little more, a little, 3 arguments a week, no more, no less, and you'll be able to have it. Yeah.
I was also thinking, Michael, of this video you recorded with George Bongard recently about how all the robots, most maybe, and AI we have now are very oriented to certainty. They're optimized for certainty. In fact, we have kind of minimization of surprise and all the other things under FEP and whatnot. While in biological systems, all of them, including singular cell, we deal with uncertainty, as you put it on that interview, on different spatial and temporal scales. And so when you have this red pop in a red paradise, it is very certain because the food is in the fridge, the roof is over your head, there's no need to fight, there's no threat. So he's licking and grooming. I'm a beautiful narcissistic rat, right? There's no other things to tackle. I think the amount of uncertainty is also playing into it. We have this term uncertainty tolerance, and if you think about AI and robots, because you looked at frustration tolerance in bubble sort, but with robots and AI, if we expose them to gradually increasing amounts of uncertainty and see what they do, I mean, that's part of the question, I guess.
Yeah. No, I think that's very interesting. Yeah. I'm slowly working on a paper I call the fog of agency, or maybe the fog of life. I don't know, but it's related to this concept of the fog of war, right? Like when stuff really goes down, it's a term that just because you lose certainty about what's going on. It's hard to make decisions. It's hard to have long-term planning. It's hard to know what everybody's doing and collate different kinds of information, right? Like all this starts to go to hell as soon as things really get tough. So that's, as you point out, that's living systems are in that state all the time. Like you never actually know what's going on. Maybe some of the systems know certain things in certain spaces, but for the vast majority of them, there are many more unknowns than there are knowns. And you have to, right? Like living things are dealing with that constantly. So I think it is interesting what happens when you try to lift some of that, and where are the optimal points. Like we've been looking at a theory of aging that you could basically summarize as the boredom theory of aging, right? Where what happens is there's a homeostatic process where you have to make the body. Well, you've made the body. Now what? Without being given novel goals, well, you upkeep it for a while, but then eventually, you start to kind of decouple. All the subsystems start to decouple slowly because there isn't an overarching goal keeping them together. So it would be interesting in those kinds. So aging, senescence, cancer, those kinds of things. What really is the optimal? I mean, we know exercise, right? Some level of tearing up your muscle tissue is good for aging, and so on. So like, can we find the optima for these things, right? For the different subsystems. For metabolic system, obviously there is one. Maybe for the immune system, I don't know. So. Yeah.
It's fascinating what you say about predictability, because in this child development world, it's one of the worst stressors, right? For a newborn or for a young child is unpredictable caregivers, for example. So it's interesting to think about that as in the context of AI, for example. It's nice.
Well, and there's another piece, fundamental unpredictability too, and actually Josh had some of the earliest work on this back in 2006. One of the things about living things coming into the world is that they don't know ahead of time where the border is between them and the outside world. And so as a brand new being, like, well, I have these things. They don't seem to be under too good control, so maybe those are mine. Maybe they're not. Eventually, you'll right. And but at the same time, you make some noises. Mom comes and gives you food. Okay, that's got to be that. I have good control over that. And then a couple of months go by, and this stuff gets better. The other thing gets a little less reliable. And so that uncertainty of what do I have exactly. So in 2006, Josh had this amazing paper with these robots that didn't know what their structure was. They had no pre-existing, so they would just flop around and eventually build an internal model of themselves based on actions they took and what happened. And so they would infer, well, I must look like this because that's the model that fits what I've been doing and what's happened to me since then, right? So they would kind of infer their own structure. And what was super cool were two things. One is they call it the evil starfish because it looked like a big starfish with like these legs. If you take off one of the legs, it very rapidly adapts because it just does the same process again, and it says, okay, well now I have fewer legs, right? So it's much more plastic. And Chris Adami wrote this amazing commentary on it, because what they found is that during periods where the robot wasn't doing anything, it was internally pruning the models and figuring out like, okay, I definitely can't be a big ball. I must be something else. And so he wrote this whole thing about how they were dreaming, that was their sleep phase, where they would sort of sort out, throw away the stuff that wasn't helpful to them, and kind of consolidate their picture of themselves in the world. It's really interesting. It's very yeah, I mean, almost 20 years ago now. Amazing.
I had one more thought of sort of what's related here is the learning part, and Australians joke, I don't remember exactly, but they said that when kangaroo wakes up, it has breakfast, goes to pee, and then has a look around. And so it's the look around business that is important. That's epistemic foraging. What's out there now that my tummy is full and everything's okay? What's out there, right? And if we look at baby doesn't learn much, the whole resources are mobilized to save life and protect oneself. You really need a safe base and secure haven, secure base and safe haven to explore. You need mommy's lap, and then the 3-year-old crawls and looks what's around. So the enriched environment means the baby feels well enough, all the needs are met, and the baby is learning a lot, and that increases allostatic complexity, right? Conversely, when there's adversity, then you're not learning as much. I don't know if it makes any sense, but I think learning is right there with this resilience. So
would love to know. See, that's very interesting. I would love to know what the physiological and transcriptional versions of this are like. So the lap story tells us what the 3D world of motility looks like, but transcriptionally, I'm a cell. I feel pretty good. My needs are met. I'm going to explore transcriptional space a little bit, right? I'm going to sort of crawl out. What happens if I crank up the BMP a little bit? No, I didn't like that. So, right, or bioelectrically too, right? Everything is good. I've settled into a nice, rhythm. Okay, can I try some things and see like how much play, and that's something that I've been thinking about a lot, is like how to recognize creative play in these other spaces. We know what problem solving looks like. We know what like hard work, and response to stressors and repair, like we know what that looks like. What does play and creativity look like in these other weird spaces? I think it would be really interesting to try to have some way of quantifying it, or at least formalizing what we're looking for.